Hello, everyone. Dr. Jack Tips here. And as usual, I'm very excited to share some information with you. We're going to be looking at the friendly viruses with this presentation. We're going to get some mastery over using four amazing products that are called bacteriophages. And you're going to be absolutely amazed at what's available and what's been available for health practitioners. Just imagine, what does it mean if when you need to deal with a nasty bacteria, what if there were a targeted product that would only go in and only take care of that particular species, or just one or two species with no collateral damage. So this is just absolutely amazing breakthrough. Hold on to your hat. We're ready to go. Our program is called Bacteriophage Comes of Age, aka the friendly virus. So here we go. We're off and running. We're going to use a little terminology here. We've got just a few words that we should know. And the first one is a virus. People can't figure out if a virus is alive or not. But I think when things have mobility and they have an innate intelligence, we might as well treat viruses like we do bacteria and so forth in the living kingdom. There is some scientific skepticism about that, but they're alive enough to make the world talk about a virus for the last 2 years and under the connotation of pandemic. So rather than talk about the very, very few bad guys that give all viruses bad names, we're going to talk about the 99.99% of these little genetic codes that are floating around ubiquitous in the soil, in the air, in the oceans, in our bodies, everywhere. We're going to look at these viruses and learn how to have more friendly relations. So a virus is simply a collection of a genetic code. And a lot are RNA, some have DNA, and they're surrounded with a coat, a protein, and then they also sometimes associate with polysaccharides. But these little bugs run around and they have some inhibition in that they don't get to replicate alone. So they're going to vicariously replicate by infecting a cell and use the components of that cell, called a host, to make copies of themselves. So these guys move around, they reproduce, they adapt, they mutate, they adapt to survive. They pretty much are functioning as little miniature packages of life code. So now let's define what we're talking about today, the bacteriophage. It's just a Greek word for a bacteria eater, and it infects and lyses, we'll come to that word in a minute, only certain bacterial species. This is so important. It takes one virus for one, maybe two species of bacteria, and it has no other way to relate to the world other than with that bacterial species. So this is a very narrow slice of life here. And bacteriophages are safe for people, for animals, for plants, for you and me. Now, there's two types of these guys. One is called lytic and one's called lysogenic, or sometimes temperate. And we get the idea. Lysogenics are going to work over time. They're not used in products or commercial applications. The lysogenic phages are not used in that capacity. So we're only really talking about the lytic species. Now, we wanted to define what lysis means. It's the dissolution or the destruction of cells. We're particularly looking at bacteria by a specific protein enzyme structure called a lysin. It'll disrupt the cell membrane. So imagine if you were in prison, wouldn't it be able to have that bucket of Roadrunner paint where you could just paint a tunnel on the prison cell wall and walk right through it and be on the outside? And so what these guys do here is the lysin is going to be released, and it's just going to dissolve the cell wall that's holding the virus in. And so this is part of their basic nature. Now, the lysogenic or the temperate phages do not cause immediate lysis, but they incorporate their DNA or their RNA into the bacteria DNA, RNA. They lay there side by side and they multiply as the bacteria multiplies. So the bacteria will divide, and then one will die. Then that virus gets loose and can do more. But in all of those multiplied species, it's a tag along. And so that presents a different scientific challenge, a different scientific understanding. So again, lysogenic phages are not what we're talking about today. We're talking about those that will lysis the cells, and we'll get more into that here. So some people are taken back by the idea of a friendly virus. Oh my goodness, I mean, virus literally from the ancient language that forms the word virus, it means poison, right? I think it's actually Latin. It's poison. And so as mankind decided to look at these guys, of course, we learned about the pathogenic species first, and they're all called poisons. But here's the thing. We got 380 trillion of these viruses in our human genome. Now, at least the last time I counted, that's how many we each have. And that's quite a few. In fact, that's more viruses than stars in our entire universe. And it's 10 times more viruses than there are bacteria. So there's a lot of these guys, and they really deserve our attention because of their numeric superiority. But you know what? 99.99% of these are beneficial. In fact, I think with science right now, there's only 128 viruses. You've heard their names like hantavirus and Ebola virus and human papilloma virus. There's only 128 of the nasty customers to date, when there's 380 trillion that maybe, if science wants to admit to ignorance and we don't know what they do, we call them commensal. And if we finally realize that everything matters, every form of life matters, every form of life uses resources and expends waste, and therefore they have an impact upon their environment. So most of these viruses are good guys, because if they were bad guys, science has pretty well wrapped up the majority of those species. So you might be thinking, friendly virus, what the heck does a friendly virus do? Well, it supports our oxygen that we breathe. Thank you, viruses. They ensure that the oxygen producing plankton, which accounts for like 90% or more of the Earth's atmosphere, and then the plants do the rest. But they have to have a nutrient base so that they're happy and healthy and busy and working their photosynthesis to the max, because that's where they're going to be releasing the oxygen and using or holding carbon dioxide. So from the dead bacteria from these guys, we're just feeding the Earth's oxygen supply. That's the basis of life on the planet. So that's what friendly viruses do. They support biodiversity. They can transfer genes. In fact, if you're familiar with like the CRISPR kind of research and things like that, we're not so much slicing and dicing up the human genome. Insertions can be made with viruses. So science is big on this subject. They maintain the balance of nature. They support certain species. They're going to multiply and keep in check species that might overproduce and become too dominant. They prevent pathogenic bacteria from overgrowing. And it's a real big role of these viruses. Listen, they protect children. Children's immune systems are too young and immature for the full range of viruses that they could encounter in the grocery store or at the fast food restaurant. And so these little phages will protect children. That's the nature's rule, like for ages 1 to 6 or so. Children's lives are dependent on these viruses. So Why don't we look at these guys a little bit here? A different and vastly superior approach to removing unwanted bacteria. So what do people do today if they have a nasty bacteria? They might go to botanicals. They might go running for a toxic antibiotic. But whatever they do, it's like, we got to kill this guy. And here we have phages or phages, however you want to say it. It's they will attack only the specific bacteria. And actually, we have been using these for 100 years. The U.S. Department of Agriculture, the grocers associations, we're eating bacterial phages with every bite of tomato, lettuce, veggies, any cut foods in the grocery store. You buy the watermelon in the little cut package, things like that. They've been approved and likely are employing these viruses to keep the bacteria in check because they prevent the spoilage. And they also inhibit the idea that a certain food could be a vector of a nasty bacteria throughout the population. So just there's 6 here in agriculture that you're already getting. You're already using these. They're already safe and approved. Salmonella, that's the phage that's used to attack the 2 species of the Salmonella aeruginosus that, we're talking typhoid, we're talking, bad stuff here, and certainly the Salmonella tummy ache. The Listeria, everyone's probably familiar with Listeria as a bad bacteria that launched Listerine and so many of the students at Lister University. The E. coli, Escherichia coli, there's some here. And Mycobacterium tuberculosis, important one. The Pseudomonas and Campylobacter. So there's 6, and there's actually more than that, but these are already being used by agriculture, food distribution networks. And so whether you know it or not, you're already a participant in biophage processes. So they are target specific. This is so exciting. If you're really so alerted as a natural health practitioner to the dangers of the bacteria. We're going to look at this some more here because, gosh, you can go in, you can address a bacteria and bring its demise and not hurt one other species. It's probably always safe to hedge the bet. 99%, you're not going to touch anything else. So here we go. Why don't we just line it up here and see what we're talking about? Antibiotics versus phage. Do antibiotics harm good species? Yeah, the collateral damage in the gut. These things are poisonous, the antibiotics. There's often tissue toxicity, tissue damage. You can have massive skin damage if you get out in the sunshine on an antibiotic, things like that. You eat some grapefruit, no telling what a person can do to really bring out the worst of an antibiotic. None of this is even a factor with phages. Antibiotics are in a cold war of escalation, of creating antibiotic resistant strains. We're going to talk further. Phages will not increase any type of antibiotic resistance. They don't cause yeast infections. They don't cause pain, nausea, vomiting in the stomach. They don't create the superbugs. In fact, the phage therapy is effective for superbugs and can help undo the massive damage that's been caused by the overuse of antibiotics in the American food supply, and often through licensed use practitioners, as well as everyone gets on the bandwagon. Whether you go to the dermatologist, you go to the dentist, wherever, they're all slinging antibiotics, and so the phages also can kill they don't kill the bacteria that actually protect us from bad viruses. So we have the bad virus kingdom, and some of those are sequestered in bacteria, and then the antibiotic kills them, and the person starts getting relations with the bad virus and so forth. They're not toxic to the body. They don't require large doses. They don't cause human deaths. And I think I was reading the other day where, I don't know, there maybe per million on Z-Pak, you're getting, it's a paltry, 60 deaths a year. So we can call that a paltry murder by Z-Pak. And it's all well and good for everybody, the millions of people eating their Z-Paks. But if you're the 60 some odd person who doesn't make it through, then it's suddenly a big deal to you. And of course, the antibiotics are poisoning the human municipal water supply because the systems don't get what people are putting into the water. Now, let's look at it from another angle here. We have, is there an antibiotic to reverse antibiotic resistant species and bring them back home? No, there's not. But the phage is yes. The phage can reverse antibiotic resistance. It can sanitize surface areas using no chemicals, very inexpensive. They actually replicate at the site of infection. So rather than taking more and more antibiotic, which is poisoning the brain, poisoning the toes while trying to get that nasty bacteria that might be bugging the pancreas or something, the phage replicates at that site, at the hot spot, and takes care of it. So we just think, my goodness, the amount of virtues of the phage are just amazing. And they're effective for antibiotic-resistant strains. So if we start looking to the phage therapy, instead of escalating, what do we do after vancomycin? Now you've got Staph aureus and the antibiotics aren't working, and the person's about ready to become a death certificate. What can be done? And here we have a humble little virus that can handle those antibiotic-resistant strains. And there's reasons for this. The antibiotic-resistant strains, they have little pumps. And here comes the antibiotic, and the bacteria just pump it right back out. They start changing. They communicate. They quorum sense. They tell others how to beat the antibiotic. That whole process is going on. That doesn't happen with phage therapy, because the virus goes inside the bacteria, completely destroys it very quickly. And there's a beautiful side effect of that phage process, and that is it avoids, the side effect is avoiding Herxheimer reactions. So see, I'm telling you, we're going to be excited to learn about these. They can pick up as the bacteria try to mutate, the bacteriophage can be right there and take care of that. And it also quickly permeates the biofilm. So the big bugaboo for the antibiotic, how do they get it through the biofilm without teaching antibiotic resistance? The phage says, that's no problem for me. And so you can see the superiority is vast. Now, if you have that questioning mind, well, why do we keep giving, you go to the doctor with salmonella food poisoning, you get an antibiotic. Why aren't they using the phage? Do you have an idea? I'm keeping my mouth shut today, but cha-ching, I have certain ideas. But again, it's a great disservice to humanity to poison with the antibiotic when there's a perfectly good phage available, approved, and being used in society. So now I just thought I'd mention, because many practitioners now are expert with Lyme disease. It's actually been the natural health people that have the herbs and the homeopathics and the acupuncture treatments and things that are helping people with Lyme and the concomitant, the Babesia, the Bartonellas. And here we have a corporation called Phage, What a neat name, huh? And they're showing that they can the bacteriophage for Lyme, which I don't know if it's that it's available. I'm letting you know it's gonna be available soon, but they're finding there's no Herxheimer reaction, and this happens. I'll see if my next slide deals with that. If not, I'll double back and talk about it. But they teach us something here from these scientific studies. 2 weeks. So if we're going to employ a bacteriophage in our practices, and someone's going to say, well, how long do I need to be on the therapy? Here's a good standard, 2 weeks. And they're coming right along with the Bartonella and hopefully Babesia, which is more the protozoas, and it's going to be a little harder to deal with. So here we go. The big point, no Herx. And we have a slight misunderstanding now, and many times we have the Herxheimer reactions from either antibiotics or botanicals because we're getting the cytokine release. It's not so much that the dead organisms are toxic, which is what a lot of people think. It's because they're weak. They're slowly dying bacteria and they're vulnerable. And then here comes the phagocyte and it gobbles up that weak bacteria that our herbs or our antibiotics have irritated. And inside our own phagocyte, they show enough gene code and organization to cause the massive cytokine response. So the Herxheimer reaction, particularly regarding Lyme, not all, but with Lyme, is the cytokine release. And now the person is having the inflammation, the swelling, they feel miserable. A truck ran over me, the body's responding almost like from the bone marrow, we get that ache, and someone goes, oh my goodness, I'm having a Herxheimer reaction. The body can't handle it. There might be a rashing that occurs from that. Here's the beauty. Phages do not cause Herx reactions. They kill the bacteria so quickly, and it goes because their methodology is to explode the bacteria. The phagocytes have fragments to clean up. And when they get the fragment, that's not enough to initiate the big Herxheimer reaction, the cytokines. So now they're just eating there. They're just cleaning up the garbage. They're not responding immunologically to a much higher danger. So I thought it might be good for us to look at there's different cycles here on this lytic cycle of what we're actually doing. But this is our mechanism. When we give one of these virus phages to a patient, the first thing those little bugs have to do is they have to go find the species that they are attuned to. So if a person had the food poisoning and has salmonella, we would be wanting to give them the salmonella phage viruses. Now, the only thing those viruses can do in the body is find that bacteria. Anything else, it's just benign, it's unaccountable. The viruses basically are asleep and just dormant, doing nothing, saying, well, gee, is there a food supply? Is there anything here to activate me? So they have to attach on, and there's little pores in the bacteria, those creepy little feet that look like landers, right? The little spider things. Well, there's only 6 of them, little bug things there landing. They're actually seeking certain crevices. So there's little feelers on those feet. They're not just a landing platform. And then there's the penetration. So, I mean, how often, we run into that with reproductive cycles, and the DNA, RNA is injected, and that's starts the process inside the bacteria. Now they start getting involved in the gene transcription, and they start breaking up the bacterial DNA. And so they just kind of go on like a wrecking ball inside a genetic wrecking ball inside the bacteria, and it's just going to fragment like crazy. It's just going to chop it all up. It's a Cuisinart for short protein gene chains, lots of little pieces. And then there's got to be some innate wisdom here in this virus, because now it starts overseeing reorganization and some replication. It's going to start reassembling pieces, and it's going to set up. And you see here in our diagram, we've got 4 new little viruses. So it's like a photocopy machine. Now, there's a limitation. We sometimes think of these viruses are going to crank out 1 million more viruses. Well, there's a limited amount of raw material. There's a limited amount of gene material in the bacteria. The science seems to think it's pretty close to 1,000. If you're just going to give it a round number, there's going to be 1,000 new viruses. So we're really running a logarithm of 1 to 1,000. And then 1,000 each produce 1,000. So what a great scale that is for moving into very high numbers to deal with any type of infection process. Even if there's a lot of raw material for the bacteria, the phage can beat it. And then in step 5, we mentioned, is they set up a conveyor belt inside that bacteria, and they're going to make their 1,000 or so of the new viruses that have no other mission in life but to go look for more of that salmonella or whatever the targeted bacteria is, and do the same thing. Little copy machines running over and over. What's cool about it, if you look at the process under high power microscopy, you actually see an internal pressure build up, and then it literally is a kaboom. It's not that these things come, popping out like little birdies from a eggshell. They explode into the internal environment and put forth their 1,000 new phages, which are now going to go and look for other, salmonellas or whatever, and do the same thing to them. So bam, bam, bam. And over a 2-week period, I think science is now saying these guys relentlessly will have controlled the environment. Of course, in the submicroscopic level like this, the universe is very vast. And so the idea that, this eradicated everything is probably not what happens in nature, but it certainly can get into, 99%. So what might be exciting for you is, yes, you can own your own biophages. So these viruses are literally right at your fingertips. And here we're going to look into that. So I mentioned for our examples the salmonella, and there is a phage product here called salvirone. And so when it's mostly the human beings, or most people are not so susceptible to typhoid anymore. But gastroenteritis, certainly, you get the food that just wasn't quite right. The chicken wasn't quite cooked. And this is why airlines and so forth, they overcook everything in those microwaves and make sure you're served completely dead food for your health and nutrition, because they don't want to risk that it might just be warmed up chicken or something here, and that, there would be a salmonella coming at someone. So it's very painful in the tummy, diarrhea, nauseous, abdominal cramping, vomiting, and so forth. So anytime I hear that someone has had a food poisoning incident, I'm like, well, the antibiotic may have shut some of it down. It may have mutated some of it, whatever. I want to get the salvirone, and I want to get these little phages to scout the terrain and make sure everything's rounded up, even post-antibiotic. Now, we all have heard about E. coli, and here we can have anything from topical skin, vaginitis, and well as E. coli in the GI tract, which is pretty famous for causing the severe cramping, as our poor model here has. And we've also heard in the last few years that there was some E. coli on lettuce and invading some fast food restaurants that were trying to have a more natural health approach and so forth. And this is where the Shiga toxin comes from that makes people so sick. And we have also the pseudovirome for Pseudomonas. And Pseudomonas is a fascinating bacteria in that people can have mild cases. And of course, I picked a few photos here for the impact of how far-ranging Pseudomonas can be. But the thing about Pseudomonas, it's very antibiotic resistant. So a lot of times if a person is only going to recommend antibiotics, you're looking at high-powered, massive collateral damage and so forth. And they justify it so self-righteously, saying, I'm going to prevent corneal ulcers and death and destruction of tissue. And that all sounds great until if we don't have the knowledge to say, well, why don't we use a phage therapy? And of course, in the agricultural and grocery industry, the food industry, this type of phage will prevent the food spoilage of which Pseudomonas would do. It makes really yucky food. So we have those. And the latest is the skin virome. We're really looking at the Cutibacteria acnes, which is a leading bacteria for getting externally into the skin pore and then causing excessive oil production, a different type of oil to be generated, clogs the pore, the bacteria have a hoedown there, and then here comes the acne pimple, and deeper the cystic acne, and now there's a phage therapy. So if people have it topically on their skin, they put this on to get some viruses on the skin that will kill that bacteria. And now the person is not having to take the disruptive synthetic liver destroying vitamin A called Accutane. They're not having to take the perpetual low dose of antibiotic that mutates everything in their body to be antibiotic resistance. Here it is, been here a long time, available for people, much better mousetrap to work with this. But look at this. There's also offerings even beyond our phage discussion. We have the bacteriophage called skin virome, but there's also dermacolonizer, which brings in some of the most beneficial probiotic species. And the two can go on topically because the two aren't going to meet. The biophage is looking for the cutiform bacteria, dermacolonizer isn't going to be affected by that. And here come the good guys. And then even as brilliant, we have the skin biome probiotic, where we really deal with the gut-skin connection. And so we can make sure there's the biodiversity that protects the skin. And so it brings it all together. I'm going to knock off the bad guys with the phage. I'm going to put the good guys on the skin, and I'm going to go in my gut, which is really where it's all about. And I'm going to make sure I've got the biodiversity of species there that directly supports the skin. And in that biodiversity of species, of course, we're going to pick up some that scientific research shows us are solutions for things such as eczemas and so forth. So people that have skin concerns have a beautiful way of addressing things completely naturally. So there you go. If we back up for a quick review, we've got the skin virome, we've got the pseudovirome, be thinking Pseudomonas. All we have to do is get the name of the species, E. coli. Again, many practitioners now are using ecovirome in relationship to bladder infections, vaginal infections, checking out that there can be prostate infections, all from the E. coli, and the bad E. coli in the intestines is also, it can produce the inflammatory molecules, the lipopolysaccharides. They can build biofilms. They can be causing chronic inflammation for a long, long time. And we have the, finally, there we go. And we have the ones for salmonella. Almost makes you wonder, gee, shouldn't I just do a 2-week run on each one and just make sure that my internal terrain is free of these pathogenic forms? And if you're pretty savvy in the gut microbiome, we know we want the E. coli species, the good guys. We want the good salmonellas. We want the good Pseudomonas. There's 2,000 species of Pseudomonas. We don't want to go in and kill the 999. Well, there's 2,000. We don't want to kill all because there's a couple of bad ones running around. So let's focus on the bad ones. How empowering is it to have absolutely targeted product like this, the virus, absolutely targeting, and everything else runs business as usual without any disruption? Is this a dream come true? I tell you, if I was a drug prescriber, I would want to be all over this. I'd sleep better at night, wouldn't you? Well, anyway, our time has been short. I hope you feel some empowerment and familiarity with these phage products and the fact that they're readily available and powerful. And we have our clinical data now. We want to make sure we're using each one twice a day. And there's some rules. You don't necessarily want to take them with maybe some of the botanicals that could be disruptive to the basic processes. So usually people will do first thing in the morning, take your phage product, last thing at night. And then, of course, if we want to employ the herbs, let's say because we know there's a bacterial issue, then they're just right there in the middle of the day. Everybody's happy. And then we can be looking at restoring the probiotics. What great empowerment we have now over the GI tract and the impact of the GI tract on the rest of the body. And right now, all we can say, I think, is number one, we want to test and use these, but we can't wait until there's more species coming. The research goes on. Dr. Shane Morris is sort of the ramrod on this research for systemic, and he's on top of many new products. They do come from very stringent scientific laboratories and much scientific research to bring these to market. We want to thank you for your attentiveness. We want to thank Systemic Formulas for providing an absolutely phenomenal set of phage products for right at our fingertips. What good work we can do. I want to thank you so much. Take care. And until next time, wish you all the best.